back to the next class in cell cycle and cell division. In the last session, you had learned about some historical aspects, basic concepts of cell cycle and the differences between mitosis and meiosis. This class helps you to understand a detailed information regarding cell cycle. What actually is the meaning of cell cycle? It is a preparation phase. It is a preparation phase before cell division. Any kind of cell, whether it is a somatic cell for the process of mitotic division or it is a germ cell, nothing but a reproductive organ cell going to forward into the meiotic cell, both cases preparation is important. So that it is preparation phase for the cell division. And this cell cycle required a lot of time. So for cell cycle and division, completely a typical eukaryotic cell in case of a human or any other animal, it takes 24 hours. Whereas in the yeast is a eukaryotic fungi which is single cell. So it required 90 minutes of time for division. So what I would like to conclude here, cell cycle and division is not same in all the organisms. The duration, processes may be same, but the duration for the preparation and division in each and every species, it is different. So if you take one cell cycle, for our better understanding, I am taking an imaginary picture here. It requires a lot of preparation phase. So that cell cycle is equal to preparatory phase plus division phase. Division phase. First one is preparation, second one is division. Together is called cell cycle. How long cell will prepare and how long cell used to divide, let us see. In case the first one, this is G1 phase. G1 phase nothing but gap 1. This is also called growth phase. So cell wants to divide means before cell wants to become bulge. How cell becomes bulge? By increasing its cytoplasm. That's why cytoplasmogenesis. Cytoplasm will increase during the first phase, that is preparatory phase. Preparatory phase is also called growth, in which three subphases are there. One is G1, second one is S, third one is G2. So in the G1, cytoplasm will increase and also enzymes and proteins were prepared. They are used for DNA replication, which must happen in the S phase. This is called S phase. What happens in the S phase? In the S phase, DNA amount will be doubled. Amount of DNA will be doubled. By the process is called replication. In second year, molecular basis of inheritance, you will come to know how exactly DNA replication takes place. Here you have to understand that in the S phase of the interphase, S phase is one of the subphase of the interphase. What happens? In the S phase, what happened? DNA gets its double amount by replication. Nothing but duplication of DNA. Duplication of DNA. Here one should understand that replication means not break. It has to be copy. Once we insert a paper in the Xerox machine, the one more copy or two more 100 or 1000 copies will come with same information. That is called duplication. So breaking means what? One paper divided into two by tearing. That is different. So duplication nothing but replication. So replication requires so many of enzymes. All these enzymes are prepared in the G1 phase. Replication takes place in S phase. Enzymes required for replication occurs in the G1. That's why it is called growth phase. In that, cytoplasm will increase. Later, G2 phase will be there. G2, so that it is a gap 2 in which cell prepare division along with the required proteins plus enzymes. So G1, S and G2, all the three together is called interphase. I phase is equal to G1 plus S plus G2. G1 in the cytoplasm will increase along with the character enzymes required for replication synthesized in the G1. In S, what happens? Replication takes place. Then why it is called S? S means synthesis. Synthesis phase. New DNA will be synthesized by the process of replication. For that, many enzymes are required. Such enzymes are not prepared in S. Enzymes required for DNA replication forms in G1. Later, the cell completely, suppose if you take the diploid cell enters, this diploid cell size will be increased just like that. Understand? Only size is increased from here to here. If you see, this is also diploid cell, this is also diploid cell, but the size of the cell is increased after the G1. Once G1 is completed, it enters to synthesis. In that, chromosome number will be doubled. Chromosome number will be doubled means what? 2n will be there, that is the chromosome number. Chromosome number is not doubling, amount of DNA will be doubled. Means if here the amount of DNA is 2C and here it will be 4C. This is the cell result of synthesis. No need to confuse, I will tell you. 2n is equal to number of chromosomes. 2C is equal to amount of DNA. When a cell is entering into the G1, nothing but it is going to be prepared itself for division. That time the condition is 2n plus 2c. The condition of a cell which enters the cell division is 2n plus 2c. Once G1 is completed, the cell status is 2n by 2c. Once S is completed, 2c become 4c. Why? Because chromosome content will be doubled, not number. Number will be same. So it will be very thickened. So that 2n by 4c happened. 2n by 4c is the feature and is also result of synthesis phase. So 2n by 4c will be entered into the G2. In the G2, all enzymes and proteins will be getting ready. Then 2n by 4c is ready for division. So during division, what happens? This all three together is called interphase. That's why from here to here, this is I phase. Cell cycle is equal to preparatory phase and division phase. Preparatory phase is also called I phase. Division phase is also called M phase. So cell cycle is equal to I plus M. Till now, you have learned about interphase. One cell cycle is equal to one interphase plus one M phase. One interphase is equal to G1 plus S plus G2. In G1, cell cytoplasm will be increased, means 2n plus 2c will be there. So cytoplasm increased but not chromosomal content. Such stage shifted to S phase. In the S phase, nothing but synthesis phase, DNA will be doubled, not chromosome number. So that chromosome number remains same, 2n. As chromosome content will be increased inside the DNA, then it will become 4c by the result of DNA replication. 2n by 4c is ready for entering the next phase, that is called division phase or M phase. Entire this is called M phase. Understand? In M phase, what is happening? I already told you in the last class. Prophase, anaphase, prophase, metaphase, anaphase, telophase and cytokinesis. So that this is called prophase, this is called metaphase, this is called anaphase, this is called karyokinesis and this is called cytokinesis. After cytokinesis, two cells comes up. These two cells are 2n by 2c, 2n by 2c, just like before said. That is why I told you, once a 2n enters and divide, two 2n cells are coming, then this is called mitotic division. During mitotic division, daughter cells are having characters just like the parental cell. No change even. Why? Because there is no random chromosomal content is occurred. As it is, copy and paste. So that's why all somatic cell cycle divisions are mitotic divisions. Advantage of mitotic division is same type of progeny or clones or same type of daughter cells are forming with the same characters like parents. Whereas in the meiotic division, along with the parental characters, some additional characters will come that we will see in the next class. So, a cell cycle consists of one interphase and one metaphase. Let us see the same concept in detail now.
We have seen in the previous video that a cell cycle has two major phases. Do you remember the names of the two phases? That's right, interphase and M phase. We have seen that the interphase lasts longer in duration compared to the M phase. The major reason being that the interphase deals with preparation for the actual division, which occurs in the M phase. Now, in this video, let's have a look at both these phases in detail. Since these phases are quite complex, we divide them in substages to understand them better. Let's begin with the interphase first. Here is an image that depicts the complete interphase in general. As we can see, the phase has 3 stages. Let's begin each stage step by step. The first stage of the interphase is the first gap phase, abbreviated as G1 phase. This is the first phase occurring in interphase, hence the name first gap phase. It corresponds to the interval between the M phase of the previous cycle and the S phase of the current cycle. Since this is the first stage of the interphase, that is, the first stage of the preparatory phase, and the phase which precedes synthesis of DNA, what do you think will be the activities of the cell? Just think about it. Well, as the cell is metabolically active, it grows in size in the G1 phase. All the organelles duplicate in this phase, and the building blocks which will be required in later stages are made by the cell. It is important to note that the G1 phase does not involve DNA replication. Now let's move to the second stage of interphase, which is the synthesis phase, abbreviated as S phase. As the name suggests, there will be synthesis of something. And what will that something be? The most crucial part of each cell, that's the nucleus, along with the genetic material within, will get synthesized, that is, duplicated. So the S phase takes up the charge of the DNA replication. Now, along with the nucleus, there is one more important organelle that gets duplicated in the S phase. Do we know this bundle of threads present in the cell? That is called the centrosome. This bunch contains smaller organelles called the centrioles placed within, usually at right angles. They are made up of microtubules which help in releasing the threads required for separating sister chromatids later. We will have a detailed look at this process in our upcoming videos. For now, all we need to know is that these threads are important for the cell division. And for this reason, duplication of the bundle containing these threads is also important. This happens in the S phase. So the S phase takes up the responsibility of duplicating the contents inside the nucleus and the centrosome present in the cytoplasm simultaneously. What next then? The next step in the process of the interphase is the second gap phase, abbreviated as G2 phase. This phase allows the cell to grow more. Along with the enlargement in the cell size, the cell also makes more proteins and organelles. The most important function of this phase is that it prepares the cell for mitosis. This phase helps the cell to prepare for the actual cell division in the M phase. So now, are we set to study the next phase? That is the M phase? No, not yet. We are yet to study one more intermediate phase. A stage which can be entered post the M phase of one cycle and before the G1 phase of the next cycle. There is a phase called the G0 phase in between. So why didn't we talk about this phase in the beginning before the G1 phase? Well, the cells may or may not opt to enter this phase. The cells may enter the G0 phase or may directly enter the G1 phase after the cell division. And what factor decides this? Well, there are several intrinsic and extrinsic factors like resource availability or nutritional deprivation which can force the cell to enter this G0 phase. But what exactly happens in this phase? This phase was classically thought to be only a resting phase, as the cell is metabolically active but does not show tendency to divide further. However, recent studies suggest different names for this phase. If the cells enter the G0 phase such that they returned to G1 phase after some time, then it's called the quiescent stage. That means the cells will be metabolically active but only arrest its further division. However, they can re-enter the G1 phase as and when possible. So this kind of phase where the G0 phase can be reversed to enter G1 will be the quiescent stage. But if the cells enter the G0 phase irreversibly, then they're said to enter the senescent phase. That means the cells will only be metabolically active but never show the tendency to divide further. Hence, this phase is called senescent phase, the one from where there is no return towards further division. So depending upon the extrinsic and the intrinsic factors, the cells enter the G0 phase, which is a metabolically active phase. The only difference is that the cells do not divide further either for some amount of time or permanently. So G0 phase is the phase where the cell division halts, keeping the cell highly active otherwise. Now that we've had a look at all the important phases in the interphase, let's have a look at the next important part of the cell cycle, that is the M phase. This could be mitosis or meiosis depending upon the type of cells. To our surprise, even this M phase shows different stages.
If we compare certain things like phase and significance and their occurrence, in between the G1, S and G2, we will have some important points. Let us synchronize them. If you see what kind of phase the G1 and S and G2 are. G1 is post-mitotic division. It's simply called. How? Suppose, if you think this is the G1, this is S and this is G2 and entire this is M phase. So what word has given here? Post-mitotic phase. So this is M means what? Mitotic. After mitotic only G1 is happening now. So that is why G1 is called post-mitotic division. Clear? Post means what? After. It occurs after mitotic division when cell is under the cell cycle. If a cell comes out of the cell cycle, so that is called G0. If cell is in cell cycle, then G1 is after the mitotic phase. Then it is also called pre-DNA synthetic phase. So in S phase, DNA will be synthesized. So G1 is before DNA synthesis. That's why it is also called pre-DNA synthetic phase. Statement is satisfied. And compared to the other S and G2 and M, G1 takes more time for the cytoplasm growth. That is why it is called longest phase. What actually happens in the G1? Cytoplasm will be increased. The same phase, if you see in the other two, that S is called DNA synthetic phase. As you know, DNA nothing but 2C become 4C. Replication takes place. Which replication? DNA duplication, nothing but DNA replication. Whereas what type of phase in the G2? G2 is called post-DNA synthetic phase. Why it is called post-DNA synthetic phase? G1 is called pre-DNA synthetic phase, but G2 is post-DNA synthetic phase. Means it occurs after DNA synthesis. Clear? That's why it is called post-DNA synthetic phase. It is also called pre-mitotic phase. It is post-mitotic phase and it is pre-mitotic phase. After G2, M takes place. After M, G1 takes place in the cycle. So this is what the addressing of different phases. Now significance. What actually happens in the G1, S and G2? In the G1, what is happening? Cell is metabolically highly active. Cell is also active here and here, but metabolically. Why? Many enzymes required for synthesis of DNA all are preparing. Preparing means cell is active metabolically. So cell is metabolically active in the G1. So enzymes for replication are prepared in synthesis in G1, and enzyme is coming from protein. Protein is coming from mRNA. So mRNA nothing but RNA. RNA, different types of RNAs and proteins are also synthesizing in case of G1. That is significance. What type of significance you can see in the S phase? Chromosome content will be doubled due to the replication. 2n becomes 2n only, but 2c become 4c. Here, histone proteins are very important for the packaging of DNA. Histone proteins are helps in DNA packaging. Newly formed DNA should pack. For that, histone proteins are required. I will tell you, proteins and enzymes and RNA required for DNA replication takes place here. Once DNA is replicated, new DNA is formed that has to be packed by DNA histone proteins. Histone proteins are helping in the packaging of genetic material. I repeat, histone proteins are helping in the packaging of DNA. Now, what is the significance of G2? G2 is another gap 2 phase in which final preparation takes for the division. What is division? M phase. M phase we will learn in the next class. So, majorly which proteins are required means tubulin proteins are required. Unless tubulin proteins are formed in the G2, M phase cannot be completed. Clear? So, now let me recap it in a quick manner.
Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis.
Cell cycle, short form is CC, is equal to interphase. Interphase short form is IP plus M phase. M phase short form is MP. Mitotic phase, simply put. So that CC is equal to IP plus MP. Now, IP is equal to G1 plus S plus G2. Then MP is equal to prophase plus metaphase. Prophase means PP, metaphase means MP, next anaphase means AP, and telophase nothing but karyokinesis and cytokinesis together. So therefore CC is equal to G1 plus S plus G2 plus prophase plus metaphase plus anaphase plus telophase. This is the final equation of the one cell cycle. Understand? So cell cycle durations are various. Longest period of the cell cycle is G1. Okay, and replication takes place, DNA replication takes place in S. Tubulin protein, which is required for the anaphase, nothing but chromosomal separation, is prepared in G2. Histone proteins are synthesized in S phase. Proteins required for replication synthesized in G1. Proteins required for cell division required in G2. So what happens in the prophase, metaphase, anaphase, telophase is our next concept. I hope you understand, guys. Go through the assignment. All the best.